give a little bit of a review, so to speak, or some sort of thing that you can actually watch over and over again because the test is coming up very soon, about the rhodopsin cycle. The rhodopsin cycle is essentially the way that your brain decides that light is happening. So this is part of phototransduction as a whole, or how we get from light to eye to brain, or the primary visual cortex, which is where all of this processing is going to kind of take place. There's some processing that happens in the eye, but we're going to ignore that for now, and we're really looking at this in a very simplified manner. Either way, when light happens, so let's say we have light. Light is going to be over on this side of things, and we have our light that's coming in. What that's going to do is it's going to make a protein that's living in these weird sacs at the end of the rod or the cone, the photoreceptor, called rhodopsin, dissociate from this other protein called retinol. And the reason for that is that retinol changes from 11-cis to all trans. So the retinol in light is all trans. You can remember that because there's an L in both all and light. So light, not light. Light, not light. And if you see what I'm doing, imagine that my body is this ring structure at the one end and my arm is the chain. So light, no light. Light, no light. Once again, when we have this retinol in its all trans form, then what happens is cyclic AMP dissociates with the channel. And if there is this cation channel that is on the surface, it is now going to be closed. That means there is no release of any type of transmitter within the cleft. And that means that the bipolar cell is going to depolarize, releasing neurotransmitter. So depolarizes, releases its neurotransmitter. The neurotransmitter is going to bind to the ganglion cell. The ganglion cell is going to send a signal to the primary visual cortex. Now that might seem a little bit weird because I didn't talk about the dark first. In the dark, and when we have our retinol in 11-cis, that means that there's going to be a cyclic AMP keeps a cation channel open. And because of that, we have a depolarization in the photoreceptor. Since we have a depolarization in the photoreceptor, there's a release of inhibitory neurotransmitter, which is going to make an IPSP in the bipolar cell, and nothing is released to the ganglion cell. Since nothing is released to the ganglion cell, there's still information that's sent to the visual cortex, but that information is that there is no information. It's weird to think about it that way, but when it's dark, you don't see anything. Should make sense. So, just to review, I lost my cap somewhere. Okay, just to review what's going on. In the light, we have our retinol as all trans. All trans retinol means that there is no longer a cyclic AMP that's associated with the channel. The channel closes. The rod or the cone no longer inhibits the bipolar cell. Since the bipolar cell isn't inhibited, that means it's activated, which then releases neurotransmitter to the ganglion cell. The ganglion cell then sends its information along optic nerve or cranial nerve number 2 to the visual cortex. In the dark, retinol is in 11-cis, and because it's in 11-cis, That means cyclic AMP is keeping a cation channel open. That cation channel will allow cations to go into the cell, which will depolarize the cell. When it is depolarized, it will release an inhibitory neurotransmitter into the cleft with the synapse with the bipolar cell. The bipolar cell is inhibited. There's an IPSP that is generated in the bipolar cell, which means that it will not send any type of message to the ganglion cell, and the ganglion cell says it's dark. So in the light, we have the top. In the dark, we have the bottom. This is the rhodopsin cycle.